we've been talking about inherited traits. Those are traits that are passed down from your parents to their offspring. We talked about Mendelian genetics and how Gregor Mendel used Punnett squares to predict how many offspring would carry a specific trait. We've used pedigrees to track a trait through a family history. And then we started talking about DNA and how DNA codes for those traits. The last part of the segment of genetics has to do with gene expression. How does the DNA code become expressed in brown eyes or black hair or attached or unattached earlobes? And that's what we're going to talk about in this lecture. We've already talked a great deal about DNA, deoxyribonucleic acid, and how it is nucleic acid, which is made out of nucleotides strung together. We've talked about the unique structure of DNA as a double helix, like a twisted ladder with two rails made out of phosphate and a 5-carbon sugar, with the nitrogen bases protected in between and sealed together with a hydrogen bond. Today we're going to talk a little bit more about RNA, ribonucleic acid. And DNA and RNA have very distinct differences that you need to know and understand. The first is a little bit less important than the others, and that is that ribonucleic acid and deoxyribonucleic acid have two different sugars. So ribonucleic acid has a 5-carbon ribose sugar, and deoxyribonucleic acid has a deoxyribose sugar instead. It's also 5 carbons. There's also a structural difference between RNA and DNA. DNA always comes double-stranded. It always has its code protected in the middle. RNA, on the other hand, is an easily readable molecule because the code is not protected in between the two rungs of the ladder. The code is exposed because RNA is only single-stranded. So the second thing that you need to know is that DNA is double-stranded and RNA is typically single-stranded. RNA also is found in the nucleus as well as the cytoplasm, but DNA in a eukaryotic cell is only found in the nucleus, and it's not allowed to leave. The third and last difference is probably the most important to what we're going to be doing today, and that is, as you know, Chargaff's rules, adenine pairs with thymine and guanine pairs with cytosine. But in RNA, adenine pairs with uracil. Adenine does not pair with thymine. RNA comes in several specific types. We're going to be talking about 3 of those types mostly today. The first is that one on the bottom left. That's called mRNA, which stands for messenger RNA. And the messenger RNA carries the code it copies from the DNA out into the cytoplasm where a protein can be made. The rRNA, standing for ribosomal RNA, actually makes up the 2 major units of a ribosome. There's the large subunit and the small subunit. tRNA is called transfer RNA, and that's because it transfers amino acids to the ribosome so a protein can be made. Gene expression happens in 2 major steps. The steps are called transcription and translation. Transcription is just how you copy the code from the DNA. Translation is where you actually make something happen based on that code. It's a little bit different between prokaryotic cells and eukaryotic cells because prokaryotic cells don't have a nucleus to protect their DNA. And so transcription and translation happens pretty quickly and pretty easily. In a eukaryotic cell, in the cells that you and I have, it's a little bit more complicated because there's that nucleus to contend with. We're going to talk about how the code in the DNA becomes a protein, which is all gene expression is. Below, you can see what happens when you transfer a gene or genetic code from one DNA molecule to another. In the bottom left-hand side, a gene from a firefly or a segment of DNA from a firefly has been transferred into a tobacco plant. The protein that's produced actually makes the tobacco plant grow. On the right-hand side, you can see a similar transformation that occurred with the pig. The pig now glows because it's carrying a gene or a little bit of DNA that codes for a protein that makes it glow as well. So a gene just codes for a protein. expressing a gene is just making a protein, I like to think of DNA as a recipe book for proteins. I know it doesn't sound as mysterious and cool as you like to imagine that DNA is, but let's pretend it's a really nice recipe book. Let's pretend it's a cookbook that's been translated for generation and generation, passed down your family history with lots of secret recipes. And that's kind of what DNA is. So of course you're going to well protect that recipe book. You're going to want to keep it for the next generation, but make sure that it's perfectly preserved. So if you had a friend that wanted to borrow a recipe out of the recipe book, you're not going to hand them the whole book. What you're going to do is first you're going to make a copy of the recipe that they want. You're not going to copy the entire recipe book. If they want a recipe for custard, you're just going to copy the recipe for custard. And then you're going to hand them that recipe, and they can take that recipe, that copy, wherever they want. And then they can go ahead and cook the custard, and they end up with the final result, which is what they wanted. So if DNA is the master recipe book, and it has thousands and thousands of different recipes, or thousands and thousands of codes for different genes, then you're going to need to make sure that that recipe book stays protected, which is why you have a nucleus. It's how we protect our recipe book. Because remember, that recipe book, it carries every recipe that you need to survive and to be uniquely you. So what happens is that when your body needs to express a protein, whether it needs to make pigments for your eyes or pigments for your skin or make a little hair or make an enzyme to digest something that you just ate, it's going to open up the segment of that book, but it's only going to open up the segment that it needs. And then it's going to make a copy. That copy is called transcription. And just like my ancient recipe book, if I was transcribing a recipe for a friend and it was originally written in a different language, I'd have to make sure that I made it a language that my friend could understand. So when DNA gets copied, it gets copied into RNA because RNA can be read anywhere in the cell. Before the RNA can leave the nucleus, we need to make sure that it has just the recipe that's needed. And so sometimes there's a little bit of RNA processing, or it clips out extra pieces of RNA that it doesn't need. Then it adds a protective cap and a tail. Not even joking. It's called the 5' cap and the poly A tail. And at that point, the transcript, or the mRNA molecule, can leave the nucleus. Once it leaves the nucleus, that mRNA molecule is picked up by a ribosome. The ribosome reads the recipes. It's stored within the RNA molecule itself, and the tRNA brings the ingredients to the table so the ribosome, which is like the cook, can put all the ingredients together in the proper order so the polypeptide, or protein, can be made. At the top of this picture, you can see a DNA molecule with several genes in order. You'll also see there's some light blue segments. There's area of the DNA in between the important recipes, or the important codes for a protein, that don't code for anything. And those are really important so that you can open up just the gene that you want to express, or just the recipe for that specific protein. So if you look at the next segment, you can see the code for the DNA and how that could be, quote unquote, read by the RNA polymerase. RNA polymerase, like DNA polymerase, adds down nucleotides along the DNA molecule. But unlike DNA polymerase, RNA polymerase adds RNA molecules, which, as we talked about, are slightly different. So going with the A, the RNA molecule would match up with a U instead of a T. The C would still go with a G, the C would still go with a G, but then that A again is going to go with a U. The A would go with the U, the A would go with the U. The C would go with the G, the C would go with the G, the G would go with the C, the A would go with the U, the G would go with the C, and the T still goes with an A in an RNA molecule. The RNA molecule is then able to go out into the cytoplasm where the amino acids are going to be brought based on the code that is read. The code in an RNA molecule is read 3 letters at a time, or 3 nucleotides at a time. And those sets of 3 nucleotides are called codons. Codons code for specific amino acids. So it would be like if I had 20 different ingredients, and I had my alphabet, and I was able to use just 3-letter words, and maybe cat stood for, of course, a cat, and then I could put dog, and that would tell me the order. I would like cat, dog, hat, right? But instead, it's not coding for things that we know. It's coding for amino acids. And if you remember, amino acids make up proteins. In order to determine which amino acid is being coded for by the RNA codon, we would use a chart like this. Now, no teacher is going to expect you to memorize all of the different possible codes and which amino acids they represent, but they will expect you to use this chart. So if you look on the left-hand side, you'll see that stands for the first letter in the codon. So if the first letter was U, and you would go ahead and you'd look at that top row. If the second letter was C, you would look at the second column. And then going down into that box, you can further look to the right for the third letter in the codon. However, I usually, once I get to that first and second letter, I can pretty much realize at this point it's going to be serine, or S-E-R, as my amino acid. If you look at this chart, it's a little bit different than the one on the previous page. However, some students like to use this chart better because you start in the middle, and you would see the top right-hand quadrant is where I would start with U. And then I would look at the beige ring, and I could find UC. And then I can also see that UCC, UCU, UCA, and UCG all code for serine. So once again, I'll find the amino acid pretty easily with this chart. I'd like you to know both charts and how to use them. Using those charts, let's see if we can decode this mRNA molecule. If you look at the first codon, it's AUG. Looking at this first chart, we would go to the third row down on the left-hand side. Then we would go to the first column, and then we would look for AUG. AUG stands for methionine. Abbreviated is MET, which is actually what we call the start codon. It's the start of every protein. You should probably know that. The next codon is AAC. Once again, as we looked at an amino acid chart, in this one we start in the bottom left-hand quadrant, go A, then look at the beige ring for that A, and then we look for C. And you can see that that stands for asparagine. Let's see if you can use your favorite chart to decode the last codon. Go ahead, pause for a moment, and flip back to the chart and see if you can decode UCU. If you came up with serine, you did a great job. Nice. So now that we figured out how to decode an mRNA molecule, how is it that a ribosome does it? Well, let's remember what's happened so far. The DNA code was transcribed or copied into an mRNA molecule. That mRNA molecule was processed and then protected and then sent out of the nucleus into the cytoplasm where the ribosomes are. The ribosomes are going to clamp down on the mRNA molecule and they're going to thread it through until it gets to methionine. Remember that start codon I told you about? Once it gets there, that's when the tRNA molecules are able to start bringing the ingredients. The first ingredient is always methionine. Now, how is it that the tRNA knows which ingredient to bring at a time? Well, the ribosome actually tries to attach lots of tRNA molecules, but the one that actually has the anticodon, or the perfect fit for the codon, that's the tRNA molecule that will be used. So the ribosome might try several tRNA molecules, But when it gets to the right one, that's when it clicks it into place and slides it to the next part of the ribosomal factory. The next part of the ribosomal factory is where the peptide bond is going to form between the amino acids. But first of all, the ribosome has to find the next amino acid in the sequence. And so it's going to try some tRNAs until it finds the right anticodon that complements the codon. That means it found the right tRNA molecule carrying the right amino acid. Once that happens, it's going to slide the mRNA molecule down, and it's going to move the tRNA molecule into the next position in the factory. When this happens, a bond forms between the two amino acids, adding to the polypeptide. As the tRNAs move through the ribosome, and once they've released their amino acid, they're released to go back out into the cytoplasm and pick up a new amino acid to try out on a new ribosome. There's a saying that the central dogma of biology is DNA makes RNA makes protein. Because gene expression, which makes each of you and I completely unique and individual, comes from the DNA molecule becoming an RNA molecule, being decoded into a protein. So transcription and translation really is at the heart of biology. Transcription, if you remember, is when the RNA polymerase opens up the DNA molecule and copies the code for a specific gene. That's called the transcript. That transcript is then perfected and polished and processed. Then it's protected with a 5' cap and a poly A tail. The new mRNA molecule that's been perfected and polished is sent from the nucleus into the cytoplasm, where it's attached to a ribosome. The ribosome reads the codons 3 letters at a time and matches it to an anticodon on the tRNA, bringing in the proper ingredients. The tRNA molecule, once it's brought in the proper amino acid, is released from the ribosome and goes out to get a new amino acid so that it can try again. All this time, the ribosome is connecting more and more amino acids into a polypeptide. Eventually, that polypeptide will be folded into a fully functioning protein, and gene expression has occurred. Whether it's going to make a tobacco plant glow or a pig glow, protein expression is really important.